moment, we will go over again the topic of chromatography, but this is one that's a little bit different because we're going to be using, we're not going to be using a column. We're going to actually be using paper chromatography. So let's talk about what those two, what that is. So chromatography, column. So remember when we separated those two dyes, that's paper chromatography, the same concept, but when we separated two dyes, that was column chromatography. So this is related, except the medium is going to be paper. So a lot more smaller scale, a lot more efficient, and it's quicker. And then these paper chromatography is usually used for analysis purposes, where column chromatography, you want to obtain the substance and separate them. So also we're going to detect molecules by staining them. So in column chromatography, there were colors that we could use to detect our compounds. In this case, we're going to detect the molecules by chemically modifying them and staining them, which will produce a visual color that tells us that what we have is an amino acid. So we're also going to be doing unknowns. So each of you will be given an unknown. So I will, at the end of this, so at the end of the presentation, make sure you stay around because I'll give you, actually for the unknown, I'll talk about the unknowns at the very end. Okay, so if I don't talk about them and I forget, please remind me because you need the unknown to complete your post lab. Okay, so calculation of RF. So in chromatography, in my paper chromatography, there's something called RF, and it tells us how far our analyte traveled up the paper or the stationary phase. So we'll talk more about that later. So we're also going to think about the structure of amino acids at various pHs. So what happens when we add a basic or more acidic medium to our solvent? How will the affinities to the paper might change? So remember, some groups have acidic and basic side chains, so that will definitely affect the polarity and how it interacts with the stationary phase versus how it gets moved up the plate through the solvent phase. Okay. So let's talk a little introduction here. So paper chromatography is a, like I said, a quick, efficient way to separate compounds for analysis purposes. So when we're doing a reaction and we want to analyze what's in it, we usually use something called, it's called TLC or thin layer chromatography, but paper chromatography fits that description as well. But in this case, we are just separating a bunch of amino acids. So the first thing we do is we load our sample onto the paper, which is our stationary phase. We put the paper with our samples on it into a chamber or a beaker, a large beaker that's covered with developing solution or solvent. That's called our mobile phase, and that's what's going to be moving our compounds through the paper. Just like in column chromatography, we use two different solvents to move the dyes through the column and collect it at the end. So same concept here about compounds that are that have similar solubilities, but they are different in polarities when we introduce them through a column or through a stationary phase. Okay, so the paper will absorb the solvent, it'll take in the solvent, causing the amino acids to move up the paper at different speeds. So the solvent will be one polarity. So the amino acids will have differing polarities based on their structures. So they will move with the solvent, but at different speeds. And then you can effectively see which amino acid is which. And we'll talk about the amino acids and how we're going to put them on the plate in the next slide. Okay. So our stain will be Hydrant. Hydrant is some nasty stuff you never want to get on you. It'll turn your hands purple because we have amino acids on our hands. So don't do that. This lab is actually, for being the last lab, it's actually pretty long because there's a wait time of around 2 hours. So if you were doing this in the lab, we would have you check out during that wait time. So it was kind of convenient because you can check out while the experiment is running, and then quickly when the experiment is done, you would just wrap things up and you would be this you'll be done with the lab for the quarter. Okay, so anhydride, it's used to stain amino acids, so it reacts with amino acids. We'll go over that later. But the RF right here is the calculation that shows how far the spot traveled up the paper. So depending on how polar something is, it'll either have a high RF value or a low RF value, and it'll travel up the paper at different distances. Okay, all right. So here's our setup. So this is not drawn to scale. So you only have 10 spots. This has like probably 20 or 30 spots, but it illustrates the purpose well. Here is our paper. We're going to mark these dots represent where you would mark the amino acid. Of course, when you mark it, it won't be black. It'll be like a colorless spot that you can't see, but you'll have to label which spot is which, so you can tell which amino acid is present in your unknown. So you would spot like a bunch of amino acids here, and then you'll spot something that has your unknown mixture. You'll spot that, and then once it's developed, the solvent will run up this plate, and this line here is where the solvent will stop, and we'll take it out and draw a line across it, and that's called our solvent front. So that's how far the solvent traveled up the plate, and then that will be useful for calculating RF values. Okay, so here is our tick marks where we label each of the amino acid or unknown where it is on the plate, on the paper. I call it plate because it just looks like a plate. If you ever use thin layer chromatography, we call them plates because they look like they have a surface that's white and a aluminum bottom. That's why we call them plates. But here it's just a paper. So just a piece of paper right here. Okay, so let's talk about this. So I encourage you to read these things because some of these things might help you answer the post-lab questions. I may have given you maybe a hint. So it's very important that you read these. Okay, we do not use pen to mark the paper because it will dissolve in the solvents, and that may cause problems. So we don't want to use pen. We use pencil because graphite won't dissolve in the solvent. So we're going to touch the paper on the sides. So when we handle the paper, we do not want to touch anywhere but the sides because the sides is where no spot will be running up the paper, and therefore it'll prevent less contamination. So you don't want to touch the center of the paper with your hands or gloves because it will contaminate and may give you bad results. So to avoid contamination, we label the spots so you know which is which, and you can later identify the amino acids in your unknown mixture. Okay, like I said before, do not touch the paper with your fingers because there are skin proteins which contain amino acids. So think about what this can mean because there's like 3 postdoc questions that ask about why you shouldn't touch the paper with your fingers, and it's going to have you think about why shouldn't we touch it and what should happen if we do touch the paper with our fingers. What should we see after we are done developing it and then later done spraying it with anhydride? So what should we see at the end? So I want you guys to think about those type of things when answering the questions in the post lab. Okay, so next we're going to do the spotting procedure. So to avoid making the spots too big, we're going to use toothpicks to transfer the solution, the amino acid, to the paper. And the reason is if you do it too big of a spot can cause potentially the spot can overlap to another lane where another amino acid should be appearing. So it could complicate your results by making it difficult to interpret. You don't want to use a too small of a spot because it might not be visible after you're done with the experiment and you're done visualizing it with the anhydride. So you want to make it kind of an appropriate size, and toothpicks are the best way to do it. So we typically, when we do TLC or paper chromatography, when we're transferring our analyte onto the paper, we tend to not use pipettes because it's hard to control the size of the spot. or the size of material being loaded using a pipette or a plastic pipette, a pasture pipette, which is glass pipettes. So usually we use something called capillary tubes or toothpicks because they transfer just a small amount of liquid that gives you the right size of the spot. So really doing these techniques in lab is an art because it takes practice. It's easy to mess up. And if you're not good at it, your results will suffer for it. Okay, so we take it by dipping the toothpick into the solution of the amino acids and spot the plate by pressing it onto the paper. We're going to spot one amino acid at a time. I know it's tedious, but remember, science is all about patience and good technique. So you do not want to rush things, so do not contaminate the solutions. All right, this part here is important. It's pretty much the last question on your post lab. So roll up the paper, but make sure the edges don't overlap. And I'll tell you why. So we're going to make sure we're going to fold it into a cylinder, then staple it at the ends, but make sure the edges don't overlap. Because if the edges do overlap, some parts of the paper will have a different flow rate of the solvent versus like the bulk of the paper. So it will affect how the solvent flows through the paper. And because some of the parts of the paper will be, kind of overlapped and kind of, they'll be kind of like you're going through more material of the paper at one specific area versus the bulk of the paper where the amino acids are. So the flow rate can actually, affect how the spots move up the plate. And it can also affect the RF value, give you a different RF value, and it can also distort how the spots look, and that can cause difficulties with interpretation. So there's a lot of variables you have to consider when, analyzing things and how to, do these type of techniques. So they're very technique, specific. There's certain things you should do and cannot do. So it's always important to establish the best technique possible. Okay, so that is why we should not roll it up into like when you roll up posters, you're not trying to roll it up where the edges are overlapping. You want to make sure you wrap it around and staple it without the edges or without any parts of the paper overlapping the other end. Okay, so that's how we're going to spot and prepare the paper for development. So now the solution is really smelly. So you will want to make sure you cover the solution with aluminum foil because it can evaporate, and you don't want it to evaporate when you're developing the paper. So you remove the aluminum foil, then you add the paper into the chamber. And then once the paper is in, you don't want to disturb the paper because that will disturb the process of the solvent running up the paper, moving the spots. So you don't want to disturb that because that can cause issues with how they appear at the end. We're going to use aluminum foil to cover the beaker so the solvent doesn't evaporate. So you don't want the solvent to evaporate because that can cause issues too with the process. So don't do that. So when the solvent has reached near the top of the paper, like, a few centimeters from the top, you're going to stop it and draw a line where the solvent stops flowing. So this is called the solvent front. So wherever the solvent stops flowing, that line I showed you earlier, that would be the distance the solvent had traveled up the paper. So then you let the paper dry and you're going to stain it with neon hydrin. Usually we do this in a fume hood, neon hydrin, and don't get any on you because it'll stain your hands purple. I think when I first hear teaching for the first time, that happened to me. So yeah, don't try. If you're ever, so students who try do this, we caution them to not get any of this on their hands. All right, so staining with neon hydrin, what is it? So it's useful because it's specific for amino acids. So when we ever do this type of analysis, there are multiple stains for different functional groups. For example, permanganate will oxidize certain functional groups. So you'll have like maybe an alcohol or a double bond. There's also other stains like stains for carboxylic acids, aldehydes, and many other functional groups. So this staining technique has many uses for organic chemistry and for identifying like what is in your compound or what is in your product. So the product is the product of reaction of the hydrogen with amino acid is colored. So it'll usually appear as a shade of purple, like purple, dark purple, kind of like reddish purple. But some amino acids will appear a different color. For example, proline, when stained with hydrogen, produces a yellow color. I am not sure why. I think proline just has a structure and causes it to have a different, causes the product to be different colored because it's a cyclic amine versus, a linear amine for all the other amino acids. Okay. So this is an example of after the paper has developed in solution. See here that I have labeled this plate. So see here, RF is the distance traveled by the spot. So the spot is right here. So you will measure, for example, you will measure from the baseline here to the center of the spot. And for example, I labeled some of these RF values here. So RF, so this will be a high RF, meaning it travels across high up the plate, whereas down here is a low RF value, so it didn't travel as far up the plate. So the two distances you want to consider is this distance here to the spot, and on the denominator is the distance traveled by the solvent. So this number will be the same for every spot because the solvent front stopped right here. So we measure this distance right here, and that will give us the distance traveled by the solvent. So you use the distance traveled by the spot divided by the distance traveled by the solvent, and you'll get a value from 0 to 1. So this, I added a lot of spots here. Of course, you'll only be working with 10, so it'll be a lot less spots, but you'll get something like this where the spots now will be visible and colored, either purple, some other shade of purple, or yellow. And then your unknown, say this was your unknown, you can compare to which lane of the amino acid it matches, and you could tell what amino acid was in your unknown. And we'll go over why it's important to include both of them on here. Okay. So we run the unknowns on the same paper with the knowns so we can easily identify what is present in the unknown mixture. If you did these separately, it might be difficult to analyze because maybe the two plates didn't run the same way, or the spots didn't proceed in the same way. Say one paper you accidentally overlapped the edges, that will cause the spots to look different for the knowns on one paper versus the other. So it'll be hard to determine what your unknown has because it won't match with the other plate. So it's always better to run it on the same plate so we can make the determination on the same plate, and that's always more accurate than saying running two separate plates. And it saves solvent, time, and resources. So there's always a lot of pros versus the cons of that. Okay. All right, so stick around. I'm going to talk about cleanup. Then it's just procedure, so like get an experience of like what you would have done if we were in lab. So cleanup: dispose of the paper, dispose of the developer solution, and wash your glassware. So locker checkout, we don't do this. We would have done it, but basically you would ensure all glassware is fine. There's no cracks or chips are found in the glassware, and any cracks in beakers or glassware should be disposed of and replaced. Wash the glassware and return it if they do not belong in the locker, such as specialty glassware. Make sure they're clean and they're not broken. And dispose of chemicals if you forgot to dispose of them after the lab for previous experiments. And then ensure all glassware is clean. Okay, So there should be some unknowns. Let me quickly open a document and I can assign them. So I'll tell you a letter and you'll just put a letter on your post lab, and then we'll be on our way. Okay, just 1 second. Okay, so there are 7 unknowns, numbers 1 through 7. So I don't know, pick your favorite number and just choose that as your unknown. And then you would watch the live video, and then you'll tell me based on the experiment that's shown and the results, what is in your unknown. So you don't have to do all 7. If you want to be, Sherlock Holmes, you can if you want, but just choose one unknown, 1 through 7. So 1, 2, 3, 4, 5, 6, or 7. So just choose one unknown number. Okay. All right, everyone, that is it for lab. Pleasure being your TA. Good luck on finals, and I wish you all the best.